Welcome everyone. Today is the second day of the third evolution days in 2021 and we together with Hilal Hoca in the first seminar of the day. And our topic will be the evolution in fungal plant pathogens. And also I want to remind you that this seminar can be counted as a Uni 101 seminar, and at the end of this seminar I will send you a link form for who wants to count this as a Uni 101 seminar. And we can start, Hocam. The floor is yours.
Okay. So thank you. Hi, everyone. So I'm Hilal Özkılınç. I've been working at the Çanakkale 18 Mart University and at the Department of Molecular Biology and Genetics. So I will be introducing to you some perspectives how evolution is important for the fungal plant pathogens, and mostly I will give to you some examples from my own research output. So before giving the main topics, I would like to give a very brief introduction about the fungi. So the fungal groups are eukaryotic groups, and if you look at the tree of life, you can see the fungi has their own specific clade. So the fungi is a fungi. So, they have very old history. It is about 3 billion years ago. It has fossil records. And also we know that almost 1.5 million species have been identified, and still every year the different species has been announced for the fungal growth. So it's very dynamic for speciation issue as well. So most of the time, once we said the fungi, mostly you're thinking about the mushrooms, and but what I'm going to talk about mostly as a plant pathogens, these are the microfungus. So you cannot see exactly, but you can see their symptoms on their hosts. So but some of them could be cultured in the laboratory, and if you culture on specific media, you can see how they looks like. And here is some examples for their cultural morphology, and you see at the top how they look like at the colony morphology, and these are the a collection of the total mycelia and spores. And if you look more details for the inside under the microscope, you can see the hypha like that, and these hypha could be these hypha are the many cells connected to each other, and this connection could be divided by the septum or may not be divided by the septum. And also every fungal groups has their own specific conidia or spores, and those are the some examples for the spore shapes of the different species of the fungi. And these fungi specifically, I am focusing on the fungal pathogens. They are using the plants as their host to live. And if you look at a very general example for a cycle, how they live in their lifestyles, if you look at the very hard conditions, mostly at the winter times, so there is not enough food or enough available host to infect, and the weather is going to be so cold and the conditions are so hard. In that case, they are keeping themselves in very specific structures, and sometimes they may go for sexual reproduction. And once they go for sexual reproduction, they will be producing a specific spores after this sexual reproduction. And these spores will be starting for the first infection at the beginning of the spring. And then after this infection, they will continue for the propagation. And in that case, they will propagate by using the asexual reproduction mode. And after these cycles and many cycles of the asexual reproduction, they will be producing lots of conidia, and whenever they find available host to infect, they will go for it. And one more thing I'm going to mention about their infection processes. Some of the fungi are known as biotrophs, so which means they infect the host, but they don't kill the host cells. So it's not a kind of mutualistic, still it is a pathogen, but it's not killing most of the time at the beginning. But some of them are necrotrophs, so they are actually killing the host cells and causing the necrosis. And some of them, the hemibiotrophs, so they can be starting as a biotroph and then they can return their cycles as a necrotrophs. What I am focusing on, the necrotrophic fungal plant pathogens in my own research. So here's some examples, what they do. Here you see, for example, on the peach fruits on the left and in the bottom, you can see how they are covered the whole fruit surface with the infection. And also another example here on the faba bean, you see the necrosis and their specific symptoms on the leaves and on the pulp and also on the seeds. So there are different fungal species can attack the different host groups. From that way, the host range also another issue for the fungal plant pathogens. Some of them have very broad host range state, which means they can attack to many different plants. But on the other hand, some of them have narrow host range, which means they can prefer only to specific group of the plants. And some of them are so host specific, which means they are almost species specific, which means they can only attack to a certain plant species, and they cannot use the host the another even closer related species as a host. So after this very brief introduction of fungal growths, now we can talk about how important their evolution. So since these are the pathogens and these are infecting the host, eventually they will be affecting the agricultural system. So we should really know about what is happening in their work, what is changing, because once we are talking about the evolution of the pathogens, which means the new species could be evolving or they evolve in view of their virulence degree, and that is also will be such a problem for management strategies, and also they could be gaining the resistance against the pesticides. From many aspects, the evolution of the fungal plant pathogens are so important, actually for scientifically also very surprising and very excited area, but also in practically very important to know what's happening. So I'll start my examples by talking about the agricultural versus natural ecosystems. So once we are talking about the fungal plant pathogens, we mostly thinking about what is happening in the agricultural systems and what are the consequences for human economy and human feeding, but also these agricultural plants actually domesticated from their wild ancestors, wild relatives, right? And they are living still in nature. And in nature, they also, what we know, and there are also many evidences host and pathogen co-evolve. So today's agricultural pathogens actually supposed to be coming from the wild systems. But even though we know that in agricultural systems, the fungal plant diseases are so major problem, we don't see such a big problem in agriculture, in the natural systems. And what could be the main reasons for that? So because one of the things, agricultural and natural ecosystems are different from each other. And for the evolutionary perspectives, we can always consider about the ecosystems, because that is very important selection pressures both for the host as well as for their pathogens. As I said, host and pathogens are co-evolved. And natural ecosystems, mostly what we saw, at least in my own research, here the research from what I am talking about on the chickpea and ascochyta systems. The chickpea, you know, it is important legumes, and it has very important consumption, and also it has a wide relatives distributed mainly in the southeastern region, and they have shared pathogens for both systems. And mostly in the wild systems, the pathogens and the hosts are kind of friendly living. There is a balance. But in the agricultural systems, because of the so much artificial selection, the pathogens also evolve against the selection pressures. What I mean, for example, applying so much fungicide is the response. The fungi is becoming more resistant to such a kind of fungicide, and also introducing so many resistant cultivars to become more sustainable, more durable against the pathogen attacks. And as an arms race issue, in evolution, the pathogens also becoming more virulent and eventually break down the resistancy responses, resistancy in the plants. So eventually what we know in agricultural and natural ecosystems provides different selection pressures on hosts, as well as it affects their pathogens. And here, for that study specifically, we detected that the pathogens from the wild host and domesticated host also genetically diverged from each other, which means these are still belong to the same species, but the populations are diverged from each other because of the different selection pressures in agricultural and wild ecosystems. So another issue about the species speciation question in fungi, that is also very interesting and a very plastic issue. So one of the things, the morphological species concept, actually there are lots of species concepts, so which is actually based on how you define the borders of the species. And here I just wrote a couple of them, but if you sort, you can find even more than 20 different species definition. So the morphological ones very commonly known, probably one of the oldest ones. So once you look at how it looks like actually in the culture and also in their spore shapes, conidia shapes, you can say which species is one. So sometimes at the very, so every species has they have very own specific characteristics, but you need to be very super expert for that one. And also today, that's of the molecular biology helps for this morphological identification to support the correct identification by producing the species-specific markers. So another thing, the biological species concept. So if that's the very general known about the species concept. If you cross two different individuals and if they are mated and if they produce the fertile progeny, then we can say that these are biologically same species. So as I said, there are the sexuality in many groups in the fungi. So they can go for sexual reproduction, and also we can cross them in the laboratory. So their sexuality is rather than being a female or male, they have carrying specific genes called mating types, and the type of these genes, we can say that's one type, that's another type, and if they have the appropriate conditions, they can go for sexual reproduction. So that is another issue, and of course, after making the crossing in the laboratory, you need to confirm the segregation, and you really get the progeny from this cross. But another issue for the fungi, one thing, there are a very big groups in fungal growth. They don't have a sexual reproduction processes, even though they carry the mating types. They don't go for sexual reproduction, even in nature, and you cannot force them in the laboratory. So in that case, the biological species concept is not going to work on that. And if you try to apply morphological species concept, still you can have so many struggles because you look at the picture in the bottom. Even if you're not expert, you can even, once you're looking at this cultures, you can say these are, looks like a difference in their morphology, how they looks like, but I can say that all these belong to the same species. So the morphological species concept is not going to be helpful all the time. So what is the solution for us? Using the phylogenetic species concept, it really helps. So by applying the phylogenetics, you can find the borders of the species. And also you can apply the species tree tools to find out if they are really separated from each other as a phylogenetic or evolutionary different species. And also very good thing about these applications, you can identify new species, and also you can identify recently evolving lineages, which means they are still not being separated as a new species, but they are giving the signals to be a new species maybe very soon. So you can identify those lineages by applying the phylogenetics. So another thing, here is the examples also from my work, and for all these speciation examples, I put some of my papers in the left below part, if you're interested. So for the last example for speciation, ecological speciation, I would like to tell you a story about the host jumping for a specific pathogen. There is a pathogen, Ascochytal lentis, actually infecting the lentil. But what we recognize, this pathogen made a host jump. So which means it changed the host, even though it was super host specific pathogen, and infected the grass seed, which is mostly used for animal feeding. And once we apply the morphological species question for these pathogens sampled from lentil and grass seed, it was detected that they are morphologically differentiated. Okay, and now we tried the crossing in the lab, and it was crossed and gave the fertile progeny, and now still biologically same species. But once we applied the phylogenetics, we weren't able to identify these two pathogens on the phylogenetic tree, so they were not evolved yet to identify phylogenetically. But in ecologically, here the ecological means based on the host preference, these two pathogens actually has the host preference, so they are preferring to infect their own host. In that case, what we said is as a species to identify as a new species this dumped pathogen, we said this still morphologically same species with its ancestor, sorry, the different species with the morphologically different species in comparison with the ancestor, biologically same species, phylogenetically same species, ecologically different species. What is the meaning of that? The species borders actually really based on researchers' definitions. So with whatever criteria you're using, you can say, according to that one, that's the species, different species. According to this definition, you can say you got a different, you identify the different species. So what else? The population genetics and phylogenetics is really helpful to understand the population structures of the pathogens, and we compare the pathogen populations, and this will give us so many information about diversity of the pathogens and the meaning of diversity. And also once you compare the similarity or dissimilarity or the dissimilarity degree could be interpreted in many ways. What are the main reasons for the similarity or the dissimilarity, the population genetic structures of the pathogens? And also we can identify the migration patterns. Also we can identify the origin of the species, and we can track the pathogens. For example, here on the based on the genetic distances of the pathogens from different countries, here the red, green, and blue colors identify the pathogens from Turkey. And here in the test parts, the pathogens from the USA, and here the pathogens from Italy. So you can see how these pathogen samples from different countries genetically close to each other or not. So that will give you an idea, and then you can use this information to make your own interpretation based on your questions. And also in a further research for the monolinear fungal plant pathogens, which are the pathogens impacting the peach and plum and cherry, sour cherry. So all these plants are called as stone fruits. So once we compare the phylogenetics, the samples from the different countries all over the world, what we see here, the pathogens are the nuclear-based phylogenetics identify more clusters, more diversity for the pathogens, but mitochondrial-based phylogenetics were showing more conserved type for the pathogens from all over the world. So here is the take-home message for you. One thing, these pathogens are not phylogenetically differentiated from each other based on the origin of the country. So the geography is not the main reason for phylogenetic differences of these clades. And another message from this representation, based on the markers you're using to identify the phylogenetics, you can resolve the phylogenetics more better, more clearly, or you may not identify enough based on the markers you're using in the phylogenetics. So the markers, the genome parts you're covering for the phylogenetic analysis also will be good enough to get a result phylogeny or not. So that's also very important what regions you choose to identify the phylogenetics issue for the plant pathogens. So another issue I would like to mention about the very interesting very interesting prospects of the genome. So genome sizes vary from species to species. So it might be from 20 megabases to 180 megabases. And the number of the chromosomes are also very interesting issue for the fungal genomes. What makes so interesting the chromosomes, in the same species, the number of the chromosomes are not equal. And there are some accessory chromosomes also called dispensable chromosomes. What's the meaning of that? So some of the species, so for some of the individuals in the same species can carry some chromosomes, but the others may not carry the same chromosomes. So these chromosomes that I am mentioning and called as accessory chromosomes are not carrying the vital genes, but they could be carrying very important genes for the pathogenic adaptation. What I mean, for example, it is known that some of the genes related with the virulence are mostly located on the accessory chromosomes. So having this accessory chromosome issue proposed the pan-genome term for the fungal genomes. What is the meaning of the pan-genomes? So there are some chromosomes, some genomic parts are shared and conserved within all individuals for a species, but there are also some other regions or chromosomes which are differ or variable among the individuals still within the same species. And also, besides this number of the chromosome issue, there are lots of structural variation are seen in the fungal genomes. What I mean is the structural variation, also one of my students, Rashid, has been working on the structural variation for the monolinear fungal plant pathogens. So there are some large insertions or deletions, and also there are lots of transportable elements occupying the fungal genomes, and all these causing structural variation inside the species for the fungal plant pathogens. And these variations could be related with the many different adaptations. So here is the specific question for us, how the structural variation are related with the fungicide adaptation for this fungal groups. So also one more thing very important for the fungal plant pathogens and also very interesting, the mitochondrial genomes. The thing I just said for pangenomics, it could be also applied as a pan-mitogenomics for the fungal groups. What I mean, so you know the mitochondrial genome. So several eukaryotic organisms have this mitochondria, and they have their own genomic, own DNA, own genomic material. And for the fungi, this genomic material, at least for the species, the monolinear fungal plant pathogens that we work on it, the individuals have their core regions carrying the genes related with the respiration process, tRNA genes, RNA genes, but also there are some accessory parts in the mitogenomes. And accessory parts including the mobile introns, non-coding introns, repetitive sequences. And here the very interesting things about the mobile introns, and this part was studied from my graduate student Gözde. And the mobile introns are elements, and these introns have two main groups, group 1 and group 2. And group 1 are carrying the genes encoding the homing endonucleases, and group 2 also carrying the genes mostly encoding the RNA-mediated moving, so for the encoding for the matrix or reverse transcriptase. So some of the fungal species are so rich with these mobile introns. So these mobile introns, why they are called as an intron, because these genes are inserted within some intronic parts, and once they are moving, they also carry these intron sequences together with themselves. And these regions are so much contribution for the mitochondrial genome expansions, and also it provides so much variation within the species. But if you ask me if there are adaptations, we don't know so far, but we are asking these questions. So here is the representation of the one of the species for monolineal axis. So you can see the mitochondrial genome map, specifically showing the mobile introns positions on the mitochondrial genome. So you can recognize here how the mitogenomes are occupied with those elements. So you can also see how they are so much found both within as well as between the genes. So another very important adaptation of the fungal plant pathogens about the evolution of the fungicide resistance, because the fungicide is one of the main chemical solution for us so far to manage the diseases and to struggle with the pathogen attacks. But of course, as a response, the fungal pathogens also can gain the fungicide resistance, and the resistance could be explored by sampling the pathogens from the environment, from the agricultural systems. Also, another important thing to know about how this fungicide resistance evolved and which generations are supposed to be required for this resistance acquiring. And if these pathogens acquire the resistance, what is actually changing in their genomes levels as a response? So for that one, experimental evolution approaches are really helpful. So that is another issue we are actually asking in my laboratory, and we are applying the directional selection to find out how fungicide resistance evolve in the pathogens and what is changed in their genomes. For example, any movement in the mobile introns or transposable element movements or any change in certain gene expression level. But here I would like to also state that such a kind of adaptation responses like the fungicide resistance or virulence, increasing in the virulence, these are all controlled from the many genes. So it's a kind of quantitative trait. And that means many regions in the genomes actually may have the contribution for this adaptation development. And it is really tough question to answer that one, but also it is really exciting even to find out in every step. So I think that's all for today from my side. So our main research for my groups, we're asking, as you may guess, speciation question, population genetics, and genetics and evolution of the virulence and fungicide resistance. And so mitochondrial genomics is one of our main interests, and so genome dynamics and the phylogenetics. And my lab is Özkılınç lab, Fungal Evolutionary Genetics lab. So if you want to have more information, you can visit us, and also you can contact with me or with my groups. And if you want to join us, you're welcome to contact. And also I would like to have a special thanks to my whole groups because doing the research is not possible by doing myself. So contribution coming from all my students. I'm so thankful and so great to work together with them. Also, I would like to thank to my financial support. And thank you for listening to me. So I just mentioned and touched to the many things. Hope it's not so confused presentation. And if you have any questions, you're going to be more than welcome.
Thank you for sharing your research. And if our friends have questions, they can raise your hands and ask their question. I think the topic was the new and for most of us, and it requires like so much further readings, and maybe they don't have questions now, but the topic was interesting and I need to read more further readings. I hope no one has question. If they have, please raise your hands, or you can write it on the chat. Okay, we can listen to Simge.
Hocam, first, thank you for your presentation. Do fungi have the different type of communication, or I just read something like that, but I don't actually certain about it.
Well, so communication, you mean how they are interact with each other? I mean,
Yeah.
specifically for within the species or between the species, I mean like the microbiota issue?
Both fungi communication, and I remember that fungi and tree communication.
Yeah, actually, I'm not so expert for that question, but what I know so far, yes, they are using a different the molecules for signaling to each other. For example, you know, there is a mycorrhizal interaction between the legumes and some the mutalis. No, that is not exactly for that one. So, but for example, what I can tell you for mating type issue, at least I can tell. So the different mating types for going for the sexual reproduction, of course, they are transcribing and producing some certain chemicals to communicate each other. But that's your question, really good question, but I don't know so much about that one. So they could be taught from the different perspectives, so how they are connecting to each other within the same species. For example, so many different isolates. For the isolates mostly used instead of using the individual term, we use isolate term. So many different isolates. So you can consider many different individuals can infect the same host. So how they are interacting in that situation, and also in the same host, there could be some other pathogens or maybe non-pathogenic fungi. So how they are interacting to each other, that's very good question, but actually I don't know exactly how they do that. But it's a good question again.
Okay, hocam, thank you so much.
You're welcome.
One of our friends asked through the chat in order to say that why did some fungi has evolved to be prokaryotes and some evolved to be eukaryotes in microbes? What are the genetic difference or the advantage of it, like in the when they like become new species? What is the advantage of this being prokaryote or eukaryote? Like, is it increase the diversity or not?
I'm not sure I get the question correctly. So the question is telling how they are becoming prokaryote or eukaryote for fungi because all eukaryote?
No, mainly ask that what is the difference? Like, what is the advantage of being prokaryote and what is the advantage of being the eukaryote for them?
What's being a prokaryote or what's being an eukaryote? It's a general question for being a eukaryote.
No, for fungi, what is the like benefits of it being prokaryote?
That's a good question. So, it's a kind of so I'm not well with the question actually.
Oh, I should
See the
ask the question again.
I cannot see.
Yeah, the question was actually eukaryotic microbe, like not about their eukaryotic properties. Their like host range. I mean, their host, like what's their target? Eukaryotic microbes or prokaryotic microbes?
Then I'm gonna pass this question. I'm sorry. But
Okay.
The owner of the question can write to me directly by explaining more. Otherwise, I would like to pass the question. Sorry.
Okay. We can later forward these questions to you, and we can also you can answer it later.
Yeah,
and
not very right now. Sorry.
No. I think, is there anyone to have question? I think no. And if no one has questions, thank you, Hocam, for sharing your research, and you separate your time for us. And thanks for the participants. I will share a form for who wants to like take this course in a seminar as Uni 101, and they should fill the forms. The university will count it as a Uni 101 seminar. Thank you, Hocam, for attending.
Okay, thank you for invitation, and thanks for everyone joining.
Thank you very much, Hocam.
You're welcome. And again, if you have still questions later on, you can always write to me.
Yeah, I'm going to mail you, Hocam. Thank you.
And also, there's also another on my talk in YouTube I did for the ecology and evolutionary biology groups. So if you want, also there is another on my talk related with this presentation related with my own work. Okay, so everyone, have a nice weekend.
Nice weekend. See you.
Bye.
Our next seminar will start at 16 p.m. Then take care of yourself until the second seminar.